Greeting to all the viewers. Welcome to Drpi Presentation. Today I am going to present on the topic Design and QBD based development of orodispersible tablet comprising co-crystallized anti-migraine drug. So let's start. So this is the executive summary. So what is the need of the project? Why this project and the vision? So what is the need? So migraine is a common disabling brain disorder and it is always undertreated and underdiagnosed. It affects the lifestyle of the patient with a reduction in social activities and professional capacity. There are attacks of moderate to severe intensity which last for 4 to 72 hours and it is one of the leading cause of global burden of disability. Why this project? So around 40% of the developed drugs and around 90% of the drugs in the development are poorly soluble. So through this experimental evaluation, we increase the solubility, dissolution rate and bioavailability of poorly soluble drug, which will lead to increase in the therapeutic efficacy and ultimately reduce the cost of the therapy as compared to conventional dosage form. The vision is to improve the treatment of acute migraine attacks and prevention of migraine. So this is an introduction. So basic introduction about migraine and CAD. So migraine, it's more than just a headache. There are various types of migraines like chronic, episodic, menstrual, with aura and without aura. Then around 90% of the people mistake migraine with headache, and nearly less than 50% of the people with migraine consult a physician or healthcare provider. So below this, you can see there is a CAD, flow chart of a CAD, that is computer aided drug design. As my work includes the use of computational tools, so I have shown the flow chart of the CAD. So how the process goes on. So here first there is a disease, then we go for target identification, then target validation, lead discovery, lead optimization, lastly the preclinical trials, and lastly the clinical trials. So at each and every stage you can use CAD, that is computational tools or in silico tools, to screen your molecule priorly at early stage itself. So we will see that further. So what are the objectives? So first is to perform pre-formulation studies of the drug with the excipients, then to prepare co-crystals of poorly water-soluble drug, to perform characterization studies of the prepared co-crystals, and to develop a formulation and characterize it. Next is selection of an anti-migraine drug. Selection of anti-migraine drug was justified based on the data obtained through in silico screening using Swiss ADME and Mol Inspiration software. So as you can see here in the table, we have compared the various anti-migraine drug along with their properties, which we have predicted from like pharmacokinetic or drug likeness parameters and bioactivity scores, which were predicted from Swiss ADME and Mol Inspiration software. Next is method of preparation of co-crystals. So we followed solvent evaporation method, in which the drug and coformer at a stoichiometric ratio was dissolved in a particular solvent and then stirred into a constant condition to facilitate the intermolecular interaction between the drug and coformer. Then the solvent is allowed to evaporate. The co-crystals were obtained and then we have characterized the co-crystals. Next is the evaluation of co-crystal, in which we have evaluated our co-crystal. First, we performed the saturated solubility study for our pure drug and co-crystals. So from the graph, you can see we have used solvents such as 0.1 normal HCl in ethanol, and the conclusion of the study is the proposed technique was found to enhance 7-fold solubility in ethanol and 4-fold solubility in 0.1 normal HCl. Then we have performed the melting point determination study using digital melting point apparatus. We have checked the melting point for our pure drug, coformer and a co-crystal. So co-crystal showed melting point of 178 to 180 degrees Celsius. So conclusion is that the co-crystal that were prepared revealed variation in melting point. Next is the analytical characterization of co-crystal, in which we have performed FTIR studies for our pure drug, coformer and co-crystal. From the spectra, we concluded that the IR bands were significantly changed in the co-crystal comparison to pure drug and coformer, and it indicates that there may be an interaction between the drug and coformer. And also the peaks were shifted slightly in the co-crystal spectra, which indicate that there is a co-crystal formation. Next is differential scanning calorimetry, that is DSC, were performed for pure drug, coformer and co-crystal. So from the peaks, we can say that the pure drug and coformer showed characteristic peak corresponding to their melting point. The co-crystal showed substantial difference in melting point compared to pure drug and coformer, and this change in the thermal properties were reported as evidence for the formation of co-crystal. Next, we performed the powder X-ray diffraction study for our pure drug, coformer, and co-crystal. Then, by looking into the diffractogram of pure drug, coformer, and co-crystal, we concluded that the pattern, the XRD pattern of the co-crystal was very much distinguishable from pure drug and coformer, and some new additional peaks were also observed in co-crystal diffractogram, which indicate that there is a formation of new crystalline phase, that is co-crystal. Next, we perform in vitro dissolution study for pure drug co-crystal and marketed formulation, and from the plot itself we can conclude that the co-crystal exhibited faster dissolution rate as compared to about 54% as compared to pure drug and 20% as compared to marketed formulation. Next, we move on towards the formulation development, where we aim to develop oral disposable tablets of co-crystals that were prepared by direct compression method using super disintegrant. And these tablets, the oral disposable tablets of the prepared co-crystals were optimized using quality by design approach, and it is further evaluated. The use of Box-Benken response surface methodology with 3 factors and 2 levels help us to found the relationship between factors and responses. So this is the final formula for our oral disposable tablet. Ingredients and their concentrations were given. So drug co-crystal, we have super disintegrant, diluent, then sweetener, binder, lubricant, glidant, etc. Next, we have evaluated our batch for pre-compression parameters and post-compression parameters like weight variation, hardness, thickness, friability, disintegration time was 45 seconds, and drug content, and all were found in range. Then we move on towards the in vitro dissolution study of QBD formulations, in which the Box-Benken design offered 17 runs. So we have performed the in vitro dissolution study of all the 17 batches. As you can see here, the plots of all 17 batches. And here we have compared our QBD batch with marketed formulation and pure drug. And from the plot itself, we can see that QBD batch shows faster dissolution rate as compared to marketed formulation and pure drug. So next is response surface plot. So this response surface plots was generated using Design Expert version 11 software, in which it shows the effect of independent variable, that's factors, on the responses. So this first one is a 3D surface plot, and this second one is a contour plot, which is also called as 2D plots. So first is for dissolution, and the below one is for disintegration time. So from this plot, as you can see, as the concentration of super disintegrant increases, the dissolution is also increases, but as the concentration of binder increases, the dissolution decreases, and it goes vice versa for the disintegration time. So it shows, it indicates good relationship between dependent and independent variables. So conclusion of my presentation is the co-crystal of the model drug was successfully prepared using coformer as a guest molecule to improve the solubility and dissolution. The co-crystal formation was also confirmed by melting point alterations, DSC changes, shift in IR bands, changes in 2 theta values in XRD. The co-crystal exhibited greater dissolution rate as compared to pure drug and marketed formulation. The oral disposable tablets were developed by Box-Benken design, and the co-crystal of the model drug possessing modified physiochemical properties were obtained and successfully formulated as a oral disposable tablets. Future prospect is the stability studies of the formulation is yet to be performed. So these are the references that I have referred. Thank you.